Thank you very much. That was so interesting. Our next speaker is John Harley. He's the founding director of the Center for Autoimmune Genomics and Etiology in Cincinnati Children's Hospital. And infectious agents, for those of you who study inflammatory autoimmune diseases, people have been looking for infectious causes of these for as long as I can remember. And he's done just some really exciting work with SLE, looking at EBV and other autoimmune diseases. So he'll discuss his EBV and autoimmunity. Thank you so much.
Thank you, Julie. I have a very interesting story to tell you. There must be about 150 idiopathic inflammatory diseases that we don't know the origin of, and lupus is one of them. This is the first image from von Heber's textbook in 1856 of a lupus patient that we, in retrospect, know for sure. She has the typical malar rash. Lupus is about 200,000 Americans, a few million around the world, women 10 times more frequently than men, 3.5-fold increased frequency in African Americans and African ancestry. We have data that I don't have time to show you that suggests that there's a molecular mimicry process happening in this disorder that leads to the autoimmunity that we see in the humoral autoimmune system that originates with Epstein-Barr virus nuclear antigen number 1. We also can show that virtually all patients with lupus are EBV infected and have done that most convincingly in children, where the rate is far lower than it is in adults. And so this virus infects, 95% of the adult human population. I guess it does not qualify for the definition of epidemic that I saw before, but it certainly affects us enormously. We usually, in third world countries and other cultures, non-Western cultures, the virus is caught in the first 2 years of life. And in Western cultures, about 70% of us are infected in the teenage years with sexual awakening. The virus is usually transmitted in saliva. It infects the tonsils and then disseminates. A few percent of people get mononucleosis, and then it lives in the memory B cells and comes out of those memory B cells slowly during life. And actually, we are suppressing, in all of us who are infected, a cancer, a EBV transformed B cell that is in latency 3 of the virus, is a transformed cell that our T cells really go berserk over. About 5 to 7% of our T cells are dedicated against this virus. And so this is just a list of the diseases that this virus causes, and I'm going to provide some evidence to suggest that there are more to this list. I will mention that multiple sclerosis is accepted, though the mechanism is not known. Mononucleosis, I've mentioned, nasopharyngeal cancer, which is kind of epidemic in the Southeast Asia, Hodgkin's lymphoma. If you are immunosuppressed as a consequence of a bone marrow transplant or any other reason why that would happen, the transformed cells that are already in you will grow into a lymphoma, and this is a really serious problem in trying to control this in immunosuppressed patients. And of course, the virus was discovered with Burkitt's lymphoma from those patients in Kenya. So in trying to understand where lupus comes from, the origin comes from either the environment, which EBV looks like it's a candidate from our perspective, or the subject. And the genetics reveal, give an indication or a tag for mechanisms that operate to create these disorders to begin with. And so the most powerful modern way to look at this are the genome-wide association studies, in which you take a large number of cases and controls and compare them and look for usually a few 100,000 markers across the human genome. The address of what exactly it is that's causing it is not precise, and so you carry in that LD block that I have there in red all those markers across, when the one that's starred in this hypothetical example is the one that's causal. And so you really don't know which one it is of that group, and so you have to consider them all. And there are now like 40,000 of these known for about almost 2,000 phenotypes in the human population. And that can be found at the GWAS catalog that you can find online. In studying lupus, we've spent the last 15, 20 years working on these genes. And that little gray line that you see down there is the point of statistical significance, 5 times 10 to the minus 8. Imagining that there's a million independent operating units in the human genome, and all of those other things that you see popping up represent individual mechanisms that are genetic mechanisms that alter risk for people who develop this disorder. And so if we know the genetics and the environment, a unified understanding of disease would require that they come together. Parsimony would be the argument that we would make since ancient Greek times to try to find simplicity, which I heard emphasized before, that would unify what we know into an idea and a conceptual construct that would then give us the power to change the diagnosis, therapeutics, prevention, and other things. If EBV causes lupus, then how do we find the mechanism? Both genes and the environment should contribute. The genetic identity changes in allele frequency with no insight into mechanism. All we really know from those GWAS studies is that one alternative at this particular spot in the genome alters risk versus the other alternative, given a polymorphism. But the GWAS studies are consistent in showing that about 90% of the polymorphisms that we find that are associated are regulatory. So we're really looking for a regulatory kind of explanation for most of these 40,000 in all the phenotypes that have been studied. And so one of the surprising things and wonderful possibilities is the virus makes its own transcription factors. And so if this is true, that the virus is important, we tested the prediction that the transcription factors from the virus would concentrate in the disease loci that we found for this disorder. Now we have about 136 genes, and when we did this study last year, we concentrated on the European group from a few years ago, which was 53. Both groups do the same thing. And how do we find that? In 2007, a procedure was invented called chromatin immunoprecipitation followed by next generation sequencing. We call that ChIP-seq. And so basically you make an antibody against the transcription factor and it immunoprecipitates, and you can see where it lines up. Where the transcription factor shares a variant with the disease is what we have here. And I'm going to show you a series of slides that look like this with the transcription, with the loci across the bottom and transcription factors across the side. When I look at Epstein-Barr virus, the cofactor for transcription, Epstein-Barr virus nuclear antigen number 2, binds half of the lupus loci. This is looking at 1,544 data sets from humans, and you can see the viral one is up there at the top, number 7, and the ones in red are already known to be transcription factors that form super enhancers in the infected B cell. And so that cluster on the left that is in the box, we would think is the regulatory cluster that is likely to be EBV related. We have evidence that the EBV infected B cell is the only cell type in which this happens, because we don't see it in B cells that are not EBV infected or in any other cell type. And then we arrive at the conclusion that a substantial proportion of the Epstein-Barr virus risk for this disease is coming from the EBV infected B cell that lives in the patient. There are other diseases that we can easily take these informatics and apply to. We have the same result in multiple sclerosis, rheumatoid arthritis, where you see EBV is number 3, the Epstein-Barr virus nuclear antigen. Type 1 diabetes, we even have evidence that in type 1 diabetes there's an EBV story that shows epidemiologic increased frequency in children. And this is type 1 diabetes, where it's number 4. Inflammatory bowel disease, it's about number 15. Idiopathic juvenile arthritis, and celiac disease. So this was published last year in Nature Genetics in the, I think, April or May issue. We can take these and now look at all diseases in which qualify for the analysis against all of the transcription factors that we can find ChIP-seq data sets. And that group that you see on the left there is the one I've just talked about with autoimmunity. And surprisingly, in the meantime, we can look at any disease. This is not EBV related, but the same kind of regulatory story happens in the 61 genes that were known at that time for breast cancer and coronary artery disease with no EBV or other viral story in this. This is the viral story. You can see EBNA2 there. It suggests that chronic lymphocytic leukemia has an Epstein-Barr virus story at its origin as a disease that wasn't included by its clustering with this. And this shows the Epstein-Barr virus nuclear antigen 3C, LP, and EBNA2 all clustering with chronic lymphocytic leukemia genes. Vitiligo is one that's come up recently, and so it's added to this group. And we can show in some instances, this is an example of showing allelic specificity consistent with the possibility that this is actually a genetic mechanism. And I don't have time to go through all this, but we think that we are adding a substantial number of disorders that are potentially causal for Epstein-Barr virus, especially multiple sclerosis and lupus, with the others probably representing minor populations. This is a virus that is, if not epidemic, it infects us all. There must be about 3% of the population worldwide that have one of these disorders that I've talked about, millions and millions of people. I need to thank all my collaborators for making this possible and all the places for supporting our research. And I see the representatives from the National Institutes of Allergy and Infectious Diseases here, for which I'm very grateful have supported me for the last 30 years.